Hi, this is our Bozeman video project for AP Biology. The cell cycle is controlled by internal and external mechanisms which can determine the fate of the cell. The cell cycle has numerous complex stages that can control the fate of the cell itself. The cell cycle contains multiple checkpoints that are essential for the occurrence of crucial events. These events happen prior to the go-ahead signal, which will be discussed later. Checkpoints are control points in the cell cycle where stop-and-go systems can control the cycle. The G1 checkpoint is known as the most important checkpoint. If a go-ahead signal is given in the G1 checkpoint, the G1, S, G2, and M phases will complete and divide. If there is no go-ahead signal received, it will exit the cycle, called the GO phase, also known as the non-dividing stage. In the G2 checkpoints, protein kinases are present. These kinases are cyclin-dependent kinases, or known as CDKs. MPF is a cyclin-CDK complex that goes hand in hand with cyclin concentration levels. The cyclin level increases during the S and G2 phases and then dramatically decreases during the M phase. The M phase is known as the mitotic stage, where the cell divides its copied DNA to make two new cells. Mitosis is the division of a nucleus, which is usually followed by the division of the cytoplasm.
Additionally, internal and external signals provide stop-and-go signs at each cell checkpoint. MPF stands for maturation prompting factor. The MPF is a cyclin-dependent kinase bound to the M-cyclin partner. The cyclins and the KDKs work together to complete cell cycle transition. The M-cyclin is minimal at first, but the levels increase near the G2 and M phase. The MPF adds phosphate to numerous proteins located in the nuclear envelope, breaking the nuclear membrane down. The MPF provides stop-and-go signals at the checkpoints, which was discussed earlier. The PDGF stands for platelet-derived growth factor, which aids in the regulation of cellular growth and division. It also provides go and stop signals at the checkpoints. The binding of PDGF molecules to BDGF receptors allows cells to go past the G1 checkpoint and divide. For example, cancer is caused by uncontrolled division of cells. The reason this occurs is due to numerous changes in cell cycle regulators. Inhibitors produce too little activity, they can aid the growth of cancer. However, if there is too much activity, this can occur as well. Cancer cells ignore the stop-and-go signals at the checkpoints, which are meant to prevent them from dividing. Cancer certainly results from disruptions in the cell cycle. Thank you for watching our presentation.